Hi everyone, my name is Caroline and I'm a postdoc at NCATS. So during my PhD, I worked with animal models to understand how drug addiction changes the brain. And the thing about animal models is that they are extremely low throughput. In other words, what might take 2 years with animal studies could take a couple of weeks using cell culture. Also, most treatments developed from these studies don't translate well to humans. In fact, while we can slow the progression of neurological diseases, most are not curable or even reversible. So perhaps one issue contributing to this problem is the way we're modeling these diseases, and it makes you wonder whether we can do better. I don't think it would surprise anyone that the brain is the most complex organ in our body, and with over 150 regions, which are each differentiated by what kinds of neurons they contain, based on the type of neurotransmitters they produce along with where they send them, it's incredibly important that disease modeling is catered to the area of the brain where the disease occurs. For instance, the prefrontal cortex and hippocampus each play different roles in memory and contain similar cell types, but in different distributions. And what we see is that diseases like Alzheimer's tend to affect the hippocampus more than the PFC. Then there's regions like the ventral tegmental area, which plays a big role in drug reward. That's one of only two areas in your brain with cells that produce dopamine. And diseases like opioid use disorder tend to have a big impact on this region. So in the 3D tissue bioprinting lab at NCATS, we are working on three-dimensional tissue culture models using human-induced pluripotent stem cells. To improve disease modeling, we have developed a method for making brain region-specific neurospheroids, or 3D cell aggregates, using already differentiated and already matured iPSC-derived neural cells. By doing this, we can control the ratios of cells present and reflect the cell type composition in different regions of the human brain, meaning that we can then loosely model the overall activity that might occur within these regions. Similar to traditional 2D models, these spheroids can be used for high throughput screening studies and made in 384 well plates, but their structure allows for better neuronal communication. We can then use a plate reader to look at the neuronal activity from each spheroid across the whole plate simultaneously. And what we found is that the activity profile is unique depending on the composition of cells within each spheroid. These graphs show the activity within our spheroids, and you can see how those containing mostly excitatory glutamatergic neurons, which are the ones modeled off the PFC and hippocampus, are more active than those with mostly dopaminergic neurons, like the one modeled off the ventral tegmental area. And since I mentioned that different neurological diseases target different brain regions to varying degrees, we can then incorporate mutant or patient-derived cell lines into our brain region-specific spheroids to more accurately model diseases and screen for potential treatments, which we believe will improve drug discovery down the line.